I think it started. Hopefully you'll have seen a message.
Not yet, no. This is what is so funny about it, that you kind of get cut off.
A message did pop up on the screen, actually.
Oh,
That's
thank you.
brilliant. Thank you.
It didn't stay there very long, though.
No. It's just very,
Okay.
very frustrating when you're sharing your screen, as I am with my slide. You can't see much else or do much else.
And the red button at the top of the
Oh,
screen
yes,
is illuminated as well.
It's going,
Yeah.
isn't it? Yes.
Yeah.
Brilliant.
Okay.
Good afternoon, everybody. Thank you for joining. We're obviously going to wait for a few minutes until everybody's joined. When you join, can you please mute your microphone and switch off your camera?
Screen.
So good afternoon, and thank you very much for joining. When you've joined, can you please mute your microphone and switch off your camera? I think we're going to have a lot of people in the meeting today, so better not to overload the system. We'll just wait a couple more minutes for people to join as they're still coming in. So 1 more minute and we'll start. Okay, so I think it's sort of subsiding a bit. So again, welcome everybody. Thank you very much for joining us. For those of you who don't know me, I'm Rosemary Collier from Warwick Crop Centre and the one who's been emailing you all. So it's a great pleasure this afternoon to welcome my colleague, Professor Gary Bending. Gary is Professor of Environmental Microbiology in the School of Life Sciences at Warwick University and Deputy Head of the school as well. And he works on the structure and function of microbial communities and has a particular interest in the plant rhizosphere, which he's going to talk about this afternoon. So the title of his webinar is Plants and Their Underground Friends and Enemies, Why Soil in the Root Zone Matters. We'll take questions at the end, but if you think of any questions as Gary goes along, then feel free to put them in the chat and then you can ask them in person if you like at the end. So I'll hand over to you, Gary.
Hi, everybody. So hopefully that worked. Can everybody see the PowerPoint?
Yes, I can.
And
Hopefully.
can everybody hear me as well?
I can hear you. Hopefully.
Great stuff. Okay. Well,
yep. Brilliant.
I will start then. So thank you very much. It's a great pleasure to be here this afternoon. Thank you very much, Rosemary, for inviting me to speak this afternoon. So what I'm going to do today is tell you a little bit about some of the work that we've been doing in my group over the past few years on the rhizosphere. I tend to be quite a broad person in terms of the science that I do, but the rhizosphere is a theme that really has been part of my career since my PhD, and I've always had projects on the rhizosphere. And so what I want to do today is tell you particularly about some projects which we've done to try to understand the composition of the microbiome of plants, particularly with focus on crop plants, although the work in my group does encompass more natural and semi-natural systems as well. So just to tell you what's coming, so what I'm going to do first of all is talk about the rhizosphere. I'm going to tell you about what it is and about its microbiome and provide a few definitions there. And then I'm going to go and talk about a project that was funded by BBSC to really try to understand how the microbiome affects plant health. And this was work that we did funded by the BBSC Soil and Rhizosphere Initiative, which was focused on oilseed rape. So big focus there. And then one of the things that I've been particularly interested in over the years is trying to understand new biodiversity. As a microbiologist, microbial systems are very complicated and there's huge diversity out there in the environment. And so a feature of my work really has been uncovering new groups, and particularly groups which have potentially important implications and interactions with plants. So I'll take you through some of the work that we've done in that area as well. So the first one then, the rhizosphere and its microbiome. So the rhizosphere really represents the zone of soil around roots, and it's a very important zone. This is some diagrams from a review that we wrote with Paul Hallett a few years ago that really tried to bring together and capture the way in which that root zone, the zone of soil around the roots under the influence of roots, impacts both the plant and is important in directing plant growth, but also has implications for the broader soil environment as well. So basically, the rhizosphere as an important zone. So you can imagine that plants fix carbon from the atmosphere and then it passes through the plants. Maybe about 40% of the total carbon fixed by the plants finds its way to the roots. And so there's huge inputs of carbon into the soil within the roots themselves. And in parallel to that, plants are taking up nutrients from the soil, particularly phosphorus and nitrogen, and then passing it up into the above ground parts of plants. So it's a hugely important biogeochemical cycling sort of area within terrestrial systems. And this figure on the left really tries to capture the importance of the root zone for the soil in these little circles, which are in yellow, and also for the plant in green. So this root zone really is important, as I mentioned, for soil carbon. A lot of carbon that actually reaches the soil goes through the roots. And of course, that's so important now for thinking about issues to do with soil sustainability and the use of the soil as a carbon store. That carbon is important for aggregation, sticking soil particles together, so providing us with soil structure, which in itself then has implications for resistance of soils to erosion and the ability of soils to store water. So that carbon also has impacts on availability of nutrients, particularly nitrogen and phosphorus, and the extent to which they're present in mineral forms or organic forms. And that carbon is also a food store for microbes and other organisms within the soil. So it's hugely important at supporting biodiversity within the soil environment. So those are sort of broader soil issues to do with the rhizosphere, but within this we also have the sort of green circles, which are about what the plant does, so what the rhizosphere does for the plant. Obviously, it's a place that roots grow. It provides anchorage for plants, and it's a place that is important for plant nutrient uptake. But of course, the rhizosphere is the place at which plants get their nutrients from as well. And it also supports a microbial population within that root zone, some of which are symbionts, which actually promote plant growth and particularly nutrient capture, and others are pathogens, which will be deleterious to plant growth. And the overarching sort of theme of this really is that these interactions, both the indirect interactions by which the plant roots affect the soil and the direct interactions that are taking place within that nutrient zone, are important for determining productivity of plants and their yield. So in terms of what that rhizosphere looks like then, plants obviously grow through the soil, and there are a number of physical processes that take place as those plant roots grow. So you can imagine that plants will compact soil, particularly at the root cap, as they grow, and that physical disturbance will affect things like gaseous exchange, water exchange, and also the microbial communities that live in that root zone environment. But plants also exude carbon into the soil itself. And it's estimated that maybe as much as sort of 8 to 10% of the total carbon fixed by the plant might find its way into the soil environment as exudates. And those exudates can be sort of really two broad categories. One is mucilage, so sticky stuff really. And you can see in this figure here, that's the blue stuff. And that's really to ease movement of the plant root through the soil. But it also aids hydraulic connectivity between the root and the soil. It aids water uptake. And plants also exude other materials into the soil, which actually have the effect of stimulating plant growth. So here is some work that we did on a BBC-funded project a few years ago, where we tried to characterize those exudates that plants release into the environment. This is some a profile of exudates that are released by oilseed rape roots, and these are characterized by NMR. And so what you can see here is we have the concentrations of various categories of compound that we find in the root itself, and the amounts of those that we find exuded into the medium surrounding those roots. And so you can see for oilseed rape, really, the dominant category of compounds that are exuded are carboxylic acids, and you can see particularly large amounts of malic acid that is exuded by oilseed rape plants, with smaller amounts of amino acids and sugar. So if when I show this slide in a lecture theatre context, I ask people if they would sort of really think about what those exudates would taste like, and so malic acid is the taste of rhubarb with a bit of sugar, so that would be quite nice. If you go low enough, if you use more sensitive techniques in NMR, you'll also find glucosinolates within those exudates, so it would taste like sort of cabbagey rhubarb, which probably isn't very nice, I don't think. But anyway, if you look deeply enough with the right techniques, you'll find a hugely complex metabolome that plants exude into the root zone. So because you can imagine that those compounds, organic acids, amino acids, and sugars, are sort of quite labile chemicals, really. And if you think about the soil as being a very complex microbial environment, you tend to find something like 10 to the 6, 10 to the 9 bacterial cells in a gram of soil. You can imagine that you will stimulate intense proliferation of microbes in the root zone. And this is really demonstrated in this picture here. The root zone is a place of intense microbial activity. And it's been suggested that there are similarities between the rhizosphere and the human gut. So both are packed full of microbes. Both have a role in providing the plant or the human host with nutrition, and both will modulate host immune responses as well. So which I think is a really nice way of thinking about the rhizosphere. It's the plant's gut effectively. So there's been a huge scrutiny over the past few years on the microbiome. If we went back about 10 years ago, probably nobody would have known what the microbiome meant. But now it's all over our newspapers. You can see TV programs that talk about the microbiome and wellness. And you can find supplements in chemists to tell you about how you should be, and supermarkets tell you about how you should be promoting beneficial microbes within your microbiome. So a microbiome really is the microbial community that inhabits and then interacts with its host. And we hear a lot in the news about all sorts of disorders that are to do with dysbiosis of the microbiome. And that really means loss or gain of microbes which result in disease. So there are lots of examples of gastrointestinal diseases, for instance, that are caused by dysbiosis. There is evidence that diabetes might have a microbiome component. Certainly anxiety and almost anything that you can think of might have an interaction with dysbiosis of the microbiome. It's really recognized that it's a very important part of our physiology. And so the same is true of plants. And so there's been a huge scrutiny, just as we've been hearing a lot in the media about the human microbiome. In the scientific world, we've been uncovering huge amounts about the plant microbiome over the past 10 years or so. And really the driver for this is the advances in sequencing technologies that have become available to us over the past 25 or so years. And so this is really a graph that just shows us how cheap it is now to sequence per megabase. That's a million bases of DNA sequence. And so this shows us in 2001. This is the time at which, you remember, the human microbiome, the human genome was being sequenced. I think it was completed in 2003. You can see that it cost about 10 million US dollars per megabase of DNA. When we get to the current era, you're talking about, 1 cent to sequence that amount of DNA. So it's a huge increase in the power of these technologies, their speed and their cheapness. And this line has sort of Moore's law, which comes from sort of general principles of when you have a new technology and how the cost of that technology will reduce the cost of using it over time. And you can see the sequencing technologies have really advanced way beyond that Moore's law. So it's a really very powerful set of techniques that we have available to us now. So just very briefly, the ways that we can use these techniques to characterize complex microbial communities, there are a number of techniques, and it can be quite bewildering, to be honest, to think about the way in which these techniques can be deployed. But I just wanted to show this because I'm going to use two techniques in the talk. So I just wanted to sort of just show you what these techniques are and how we can use them. So the central issue with these techniques is that we now have the possibility of sequencing nucleic acids. So DNA and RNA. DNA will tell you about the nucleic acids that are present in a particular system. Now, this might be telling you about nucleic acids that are present in living organisms, but because they can be released into the environment, it can also tell you about dead organisms. So if you're interested in archaeology, for instance, when people are looking at genomes of people in cemeteries, they will be sequencing DNA they've extracted from those bones. So DNA can be quite persistent in the environment. If you want to find out about the active communities, those things that are live and working at the moment, you can extract out RNA. And so this will just tell you about active pathways, active organisms within a microbial community. So you have these, first of all, the choice of target. Do you want to know about the active communities at that particular point in time? Or are you more interested in a broader set of organisms that inhabit that environment? And then we have a number of approaches that we can use once we've extracted that DNA or RNA. Now, the most basic technique, and the first technique really to come available, was something called metabarcoding. And this is something that you would do with your DNA to target one particular gene. And so we have a variety of genes that we can use as targets that will profile, for instance, bacteria or fungal communities, or within those, if you're interested in pseudomonads, you can also find a metabar technique that will tell you just about that particular microbial group. And so what you do is you take that one gene and you amplify by PCR lots of them. And then you can look at the gene sequences that you have and annotate those gene sequences against databases to tell you which organisms you've got in your community. You can also use that in a functional capacity as well. So if you're interested, for instance, in denitrification, so loss of nitrogen from the soil as a gas, you can actually look at denitrifying genes and find out about the types of denitrifying genes that you have in your system and the microbes that are contributing to that particular function. So it's a very cheap, very powerful technique. But more recently, what we have the power to do now is, rather than just targeting one particular gene, we target all of the genes contained within the community. And this is a technique called metagenomics. And so you take out all of your DNA, you sequence it all, and you can either you can pull out particular genes within that metagenome that will tell you about functions. And now you can tell all functions. It can be nitrogen cycling, phosphorus cycling, pathogenesis. You can also assemble those genes into genomes. And so then you can work out what viruses you had in your sample and what was the genome complement of individual viral genomes, for instance. And you can do the same techniques, metabarcoding and metagenomics, with RNA. In the case of RNA, when you're sequencing at that community level, you're then comparing metatranscriptomes, so the active genes within that community. So hopefully, and get some idea from that about the sort of techniques and their adaptability for studying complex microbial communities, whether they're part of a sort of human microbiome or the environment. Okay, so that's the rhizosphere then, and that's microbiomes and how we study microbiomes. What I want to do now is tell you about some work that we've done to try to understand the microbiome of crop plants and how that might affect plant health. So the way that we got interested in this was some work that we did, funded by DEFRA actually quite a few years ago now, with a phenomenon called yield decline. And really there's evidence, a lot of evidence out there in the literature, that if you grow a crop under short rotations or in monoculture, you lose productivity. And this is some data from a literature review that Amanda Bennett did when she was at Warwick. I think Amanda's now at AHDB, and she came to do a project with Dave Chandler and Peter Mills, John Whips and myself, which was about the phenomenon of yield decline. So she pulled together data from the literature to try to find, what were the implications in terms of yield loss from growing crops in short rotations or monoculture. And she collated it, and you can see some of the data is here. So you can see for wheat, there's evidence that monoculture or short rotation can reduce yields by 9 to 20%. And you can see that for some crops, there's evidence for huge yield losses through these monocultures or short rotations. You can see rice here up to 59% loss of yield, and sweet potato and sugarcane over 50% loss of yield in some instances. So part of the review really was bringing this data together to try to understand what was the scale of this yield decline and try to understand what the factors driving it were. So you can see this figure here on the right of your screen, which shows that really this yield decline can be affected by site-specific factors like the particular cropping history that you have, soil type, moisture conditions, climate, but also the management practices that you have at your site, such as, for instance, whether you use tillage or pesticides or the fertility treatments. And we try to bring together the sort of the suggestions, what has been suggested in the literature as being responsible for this yield decline. And the important thing about this yield decline is that usually there isn't a particular pest or pathogen associated with it. When you look at the pests and pathogens within your crop, you can't really see anything wrong with it. It doesn't seem to be because of a pathogen. And so really the suggestion is that this is because of dysbiosis of the microbiome. And this is what we built this particular project about, to try to understand this yield loss and try to understand it in the context of dysbiosis. We focused on oilseed rape because it's an important UK crop, and this is just some data on values of the crop. You can see that it has a huge value in the UK and globally, 72 million tonnes globally, just in the UK, worth almost £1 billion. And of course, it's important for not just because of oils, but because of biodiesel now and an use of animal feed. And this is some data that we really sort of was really happening when we had this project with Amanda. This is some data from a project at NIAB TAG with Velcourt, which is really to do some very nice experiments to try to understand what the impact of different rotation periods of oilseed rape was on yield of the crop. And this is in a sort of commercial context. And what we have here is that a very, very nice experiment. So you had virgin, so the first time oilseed rape had grown in a particular location, oilseed rape grown after 1 year in 3 with wheat, alternate with wheat, so every other year with wheat, and then continuous oilseed rape, with all of these rotations available in a single year, so they could be compared. And the data really showed us that when you grow oilseed rape as a continuous rotation, you can get yield losses of 25 to 30%. And you can see the more intensively you grow oilseed rape within your rotation with wheat, the greater the yield decline. So with a 1 in 3 loss, you might have 10 to 15% losses, for instance. And then when you equate this, when you look at the way in which oilseed rape has grown across the UK, in terms of these rotations, DEFRA keeps data on this, it equates to a loss of maybe 43 to 86 million pounds just in England and Wales. If you extrapolate this globally, there are huge losses from this yield decline. And so really, we thought this was about microbiome dysbiosis. And so the project, really, we got some funding from BBSC in this Sarissa initiative. And it was really, we were asking a number of questions in this project. First of all, which microbes live in the oilseed rape microbiome? And then what factors shape the microbiome? What controls which organisms live in the microbiome of oilseed rape? What are the roles of soil type, climate, and the way in which we manage the soil and the crop in determining crop composition? And then thinking about the assembly of these microbial communities, how are those linked to crop health? And particularly trying to understand how these microbes are linked to crop yield decline. So the way that we did this, and I should say that we published this work a number of years ago now. The way that we did this is we went to collect samples of oilseed rape from commercial fields in the Velcourt network. So Velcourt were a collaborator in this work. And on the left, you can see the locations at which we collected samples. We had about 37 different fields that we collected samples from. And you can see by comparison to the right-hand figure, we were really taking these from the major oilseed rape grow regions in England. The way that we did it is we went out the green bud stage and we collected plants and brought them back to the lab. The important thing about collaborating with Velcourt is that because they have an excellent way of retaining information about cropping history within their fields. So we're able to actually get very detailed agronomic information about the variety, the field cropping history, the soil type, the way in which nutrients and fertilizers have been applied to the system over the years, and things like tillage history. And we can combine that with collecting data on climate. We also collected soil from these locations and did a full soil analysis for things like carbon content, nutrients, pH, and so on. We brought these roots back to the lab, and then we collected 3 fractions, really, to understand the microbiome. The first was bulk soil. So this is soil that didn't have roots growing in it from the field. We took the plant roots that we collected in the field, and then we washed them. And that washing process removed the tightly adhering soil, which represents the rhizosphere soil. And then we also have the roots left behind, which are cleared of soil. So it represents the microbes that are living in the root itself. We extracted DNA from these fractions, and then we sequenced the DNA. And we used amplicon or metabarcoding sequencing to do this. And you can see that we profiled bacterial, fungal, and protist sequences. And you can see these are the numbers of sequences and species, really, called OTUs, operational taxonomic units, that were able to profile over the systems. And this really gives you an idea about how complex the microbial communities in these cropping systems were. So we had about 14,000 species, if you like, of bacteria, almost 6,000 species of fungi, and 2,000 species of protists within these crop fields. Using the metadata, we first of all looked to see whether crop yield was linked to rotation. And it was strongly linked to rotation. So this is a highly significant relationship, and it followed exactly the same trends that we saw in the controlled experiments from NIABTAG and Velcourt. So the more intensively you grow oilseed rape, the greater the decline in yield within these fields. So the first thing that we did in terms of looking at the microbiome was look to see how composition compared across compartments: root, rhizosphere, and soil. And so that's what's displayed here. We have the 3 microbiome targets: the bacteria, the fungi, the protists. The root profile, the fingerprint of the microbial community, is shown in blue for the roots, green for the rhizosphere, and red for the bulk soil. And basically, each of these different points represents a single field within, or a single sublocation within a field. We have 5 sublocations per field. If the points are close together, it indicates that the composition of that community in those 2 sampling locations is very similar. If they're very far apart, it indicates that they're quite different. And so you can see that the blue root samples are clustering together, and they're different to the rhizosphere and the bulk soil. So we have a distinct population within the roots, a distinct population in the rhizosphere and the bulk soil. The root samples are quite spread out, particularly the fungi and the protists, which is indicating they're quite stochastic, so a lot of variability in those samples, which I think is quite important. We're then able to look at those sequences and say a little bit more about the taxa that we have within the microbiome. And that's what we're showing really in these bottom figures. They're quite complicated, but I just want to illustrate what we have is the percent abundance of, on the left-hand side, bacterial phyla, and the colours indicate the types of phyla that we have in those samples. So you can see going from bulk soil to rhizosphere to root, we can see we can have more pink, which means that proteobacteria are particularly inhabiting the root. They're enriched in the root. If we look at the fungi in the middle, you can see the green is becoming particularly enriched in the root. So as we go from bulk soil to rhizosphere to the root, we have a lot more of the Chytridiomycetes in green appearing. And we have a similar story with the protists, but perhaps not as striking as we have within the fungi. So the first question really, I won't tell you a lot about this, but just to think about what controls microbial community assembly within these root, rhizosphere and soil compartments on the landscape scale. What we found is that the most important factor shaping the composition was actually pH. This was important no matter which microbial group we looked at and no matter which compartment we looked at. We were particularly interested in rotation frequency because this is what's driving the yield decline. And we found that the only associated frequency was actually fungi. And you can see that that was accounting for maybe 5% of the variation in communities across our samples in the rhizosphere compartment and about 11% for the root zone. So it's suggesting that maybe yield decline, maybe this impact of rotation on dysbiosis was to do with the fungal community. So I'm really going to focus today on telling you about the fungal community. And I can see this is very complicated, but I just want to illustrate what we have here at the next level of resolution of our microbiome. And this is really telling us now about the specific species that we have within our microbiome in the bulk soil on the far right here, in the rhizosphere and the root. So what we have here on the left is the different species that we have, starting off with a fungus called Opidium brassicae. At the end here with relative abundance, we can see its relative abundance in those 3 compartments. So this top one here, Opidium brassicae, of the total abundance of fungi that we have in the root, 52.9% of that abundance was Opidium brassicae. So it's over half of the sequences that we got back were Opidium brassicae in the root. And you can see that it's incredibly enriched in the root compared to the rhizosphere, which itself is more enriched compared to the bulk soil. So if you look down the list, you can see that those organisms which were particularly enhanced in the root zone are in red. And so you can see we have a number of taxa there that were enriched in the root zone. We have a number of taxa that really liked it in the rhizosphere. So you can see 2 OTUs here, OTU 9 and 17. We called them this because they really don't show much similarity to database entries, so they're quite novel taxa. And you can see these particularly like the zone between the root and the bulk soil. And you can see other organisms here that really like the bulk soil, and they become deselected as we move towards the root zone. So they favour the bulk soil. And so you can see that there are a number of Opidium brassicae sequences. Gibelliolopsis likes the bulk soil. You can see that there's a tetracladium here that also likes the root. So a number of taxa there that seem to be becoming enriched in the root zone. Once we have a name on these organisms, we can actually infer what they might be. Are they pathogens? Are they mutualists that support plant growth, or are they saprotrophs that might be eating organic matter within the root zone or the soil? And there's a particular program that we can use to actually take those taxa and assign them to what we call a trophic mode. So is that fungus called a pathotroph or a pathogen? Is it a saprotroph? or is it a symbiotroph, so a sort of a mucus like a mycorrhizal fungus? And that's exactly what we did with all of these sequences. But what we did is split up our samples as short rotation samples, medium rotation samples, or long rotation samples. The short rotation samples will be alternate or continuous oilseed rape within our rotation. The medium will be 3 to 5 years between crops of oilseed rape, and the long rotations will be 6 years plus in between oilseed rape crops. And so just to show, it's quite complicated, but just start up here on the left. This is the data that we have for bulk soil. And if you look here, you can see that we are finding pathogens in the soil. There's no real enrichment between the short and the long rotations. And most of the organisms that we have within the bulk soil samples are actually saprotrophs. If we move into the rhizosphere, you can see what we have here now. We have more pathogens. So you can see the green here is approaching maybe about 30% of the relative abundance of the community in the short rotation. And you can see there's a bit more of the pathogen within the short rotations than the long rotations. And then when we look at the root itself, you can see now we're seeing about 70% of the sequences that we got back are actually pathogens in the root zone of short rotation samples. And you can see that's elevated relative to those long rotation sequences. And so really it's suggesting that short rotation is actually collecting pathogens. There are more pathogens as we have a shortened rotation within the roots themselves. And then when we looked at what those pathogens would be, we found a number of pathogens that were enriched in roots. I've mentioned Orpidium brassicae already. We also found Pyronikita, Fusarium, Pyronopisiza, Itacinelia, Phytophthora, Gibellulopsis, and an oomycete called Legina radicola. So a whole range of pathogens that we found in the roots. So the question is, which of those pathogens might be associated then with the yield decline? And the way that we did this is very complicated, so just bear with me. This is the microbial interaction network. So what we did is we looked for correlations actually between rotation and yield. So remember, we measured yield from these locations, and we correlated different taxa with both rotation and yield. And this is what one of these microbial interaction networks look like. You can see here, hopefully you can see my cursor. We have rotation here. So these are all taxa. If they have a B, they're bacteria. If they're F, they're fungi. If they're P, they're protists that were correlated with rotation and similar correlated with yield. If they're red, it means there was a negative interaction. So this one here, F2, this was negatively associated with yield. So when it was in high abundance, there was low yield. And it was also present in high abundant with shortened rotation. So F2 is associated with rotation and yield. And when we look at what F2 is, it's Orpidium brassicae, that organism that we have in very high abundance within our roots. And so this is really telling us that it's a signature of dysbiosis, and we think it's the cause of the yield decline. And in fact, in the earlier work that we did funded by DEFRA, we actually were able to isolate Orpidium brassicae, add it back to oilseed rape, and we found that it did have a negative effect on the growth of oilseed rape. So really, this is probably explaining what we have in terms of our yield decline. It's enrichment of pathogens across the landscape, across all of these 37 different locations. Orpidium brassicae seems to be the big driver of yield. But of course, we have all of these other pathogens associated with oilseed rape, and these are usually locally abundant. So they will be affecting yield at individual locations and maybe multiple farms within that network, but they're not important at the landscape scale of providing us with a decline in yield. So we were able also to look at organisms which were positively associated with both rotation and yield. And so we can see a number of taxa here. So this one, F108, again, a fungus, F19, positively associated with both yield and with rotation frequency. And when we looked at the identity of these organisms, all of these organisms here came out as being a fungus called tetracladium. So really this is telling us that this organism is a signature of a healthy microbiome. So the question is it a beneficial organism, or is it a signature of a community that is not impacted by Olpidium. And so we haven't really got to the bottom of that bit yet, but I want to tell you a little bit more about this tetracladium because we're really uncovering quite interesting biology. So when I was an undergraduate, the interesting thing about tetracladium is that it was named as an aquatic hyphomycete. And this means that it's a fungus that lives in rivers and it decomposes leaf litter within rivers. They've got this really amazing tetraradiate spores here. And this is supposed to be an adaptation to allowing spores to move through the aquatic environment. And these sort of protrusions make it stick to litter as they find litter, stick onto the litter, and so they can grow on the litter, find new litter to exploit and grow on as a saprotroph. So it was really quite interesting to find this organism as a root endophyte. And so we spent some time trying to understand this in a little bit more detail. And so one of the ways that we did this, and this is a bit busy, sorry, but I just want to illustrate some of the ways in which we tried to do this. This is from a PhD project by Anna Lazar in my group. And so what we wanted to do was really try to understand more about the sort of landscape distribution of tetracladium species. How diverse are they? What habitats they live in, what controls their abundance within the soil. And this is some work that we did with UKCEH using their countrywide survey set of samples, where they have 1,000 samples of soil from across the landscape, from all of these different vegetation types, including crop systems through to heaths and bogs, grasslands, woodland systems, to try to understand where the tetracladium was located. And you can see that these circles show locations where we have abundant tetracladium. The size of the circle shows us the color shows us the habitat. The size of the circle shows us the abundance of tetracladium within the fungal communities in those locations. And you can see the tetracladium really is abundant in these green systems, which are the agricultural systems. So really, these tetracladiums love agricultural soils. As part of this, we're really able to uncover the diversity of tetracladium. This is the phylogenetic tree that shows the diversity of tetracladiums that we found in the samples of soil from across these landscapes. And you can see there's a number of different colors here which indicate different species of tetracladium. Now, what we found is that a number of these tetracladiums we'd also found in aquatic systems, and these are indicated with a raindrop here. So tetracladium maxilliform, tetracladium furcatum, tetracladium marcellanum, tetracladium apiense. These are all well known from aquatic systems. But in soil, we had a whole range of new tetracladiums that are new to us that we didn't know about before. And you can see here, tetracladium group 1, very abundant group, very diverse. It was also apparent from the work that we did that only a small subset of these tetracladiums liked to live in association with roots. This one here, tetracladium maxilliform and tetracladium furcatum. So these are really the indicators of microbiome health, not the wider tetracladium group. The other thing that we did is to isolate tetracladium and see how it impacted plant growth. And unfortunately, we weren't able to show any positive benefits of tetracladium to plant health. But what we did find is that when we grew it with plants, it tended to stick around the outside of the plant root. This is a fluorescence microscopy image here where we grew tetracladium. We labeled it with a fluorescent tag and looked to see where it was located within the root zone. And really what we found is it was located on the surface of the root itself. We also sequenced the genome of tetracladium. So this is particularly a root endophytic strain of tetracladium, and we looked at its genome. We found no signatures of biotrophy, indicating that it's dependent on its host for its nutrition. We found that it was really great at degrading plant cell walls, particularly pectin and cellulose. And we found that it also had loads of chitinolytic enzymes, like really very abundant. And so really from its location, really forming a sheath outside the root, having a lot of chitinases, we think it might potentially have a role in fungal antagonism. Maybe that's what it does within the root. It's able to antagonize fungal pathogens. Okay, so that's tetracladium then. So I want to move on now. This is obviously telling us about novel biodiversity. There's a lot of really interesting stuff that lives in the root zone. You can imagine that with that tetracladium, it was new to find that. Really, you can still see that it could be important. You know, you can imagine tetracladium being a signature that we can use. It may be also something maybe as an inoculant that you can use. And so there's really the potential there to exploit that rhizosphere biodiversity into the future to support cropping systems that maybe reduces our reliance on fertilizers, reduces our reliance on crop protection products. And so I really think it's very important to try to understand that biodiversity. And so I really want to take you now onto another project that we've been doing, another theme really, to understand new rhizosphere biodiversity that might have sort of potential into the future in terms of low input cropping systems. And I'm sure that you will be familiar with arbuscular mycorrhizas. And this is something that I've been interested in for a number of years, really. If you know about mycorrhizas, you'll know that there is symbiosis between fungi and plants. There is symbiosis in which the plants trade carbon for nutrients. The plants fix carbon for photosynthesis. They trade it for nutrients, particularly phosphorus, from the fungal partner. Now, the fungal partner exists within the soil as spores, and this is a nice image of some spores collected from soil. You can float these out with sugar, with sucrose, and you can characterize sort of spore morphology to tell us about the species that you might have in your sample. These spores germinate when a root grows near them. They grow in, they penetrate the root, they grow through the root, they form these arbuscules, Greek for many trees, within the roots. And this is a close-up of an arbuscule here. You can see a huge surface area for nutrient exchange, which takes place in these arbuscule structures. And then within the soil itself, you can see that these fungal hyphae, they grow from the root into the soil surrounding the root. You can see they move beyond this root depletion zone, phosphorus depletion zone. Phosphorus is a very immobile nutrient in soils. Once plants assimilate it, it doesn't diffuse back into the root zone. So these fungi grow beyond that depletion zone and access phosphorus from places that plants can't access this phosphorus. Now, the arbuscular symbionts, they're obligate symbionts. They cannot grow in the absence of a host. They can't be cultured. And what we find is that they tend to be depleted in agricultural systems, adding fertilizers, tillage. These are the things that really reduce abundance of these mycorrhizas in cropping systems. We know a lot about the diversity of these arbuscular fungi. Most of the understanding that we have comes from this glomerales group, but we also have sort of arbuscular fungi in these divisions, Archaeospirales, Paraglomales, Diversospirales. But the important thing is that we assume that arbuscular mycorrhizas are some part of the subphylum Glomeromycota. And this has been something that we have assumed for many decades, that arbuscular mycorrhizas are formed by Glomeromycota. However, oh yeah, and first of all, tell you why we should be interested in mycorrhizal fungi. So the reason is because they have the potential to promote crop yield. This is some data from Marcel van der Heiden's group in Switzerland. And what they did here is they took one particular mycorrhizal fungus called Rhizophagus irregulare, and they applied it to 54 maize fields in Switzerland. And you can see they did this over 3 different years, 2018, 2019, and 2020. And they looked at the response of the maize crop to inoculation with this Rhizophagus. And you can see that there was a whole range of responses. Here on the left, you can see some fields which were very responsive to inoculation with this mycorrhizal fungus. And you can see that in this field here, you've got a 40% increase in yield when you added this fungus. And you can see lots of other fields here where you've got a response. They called them high mycorrhizal growth responsive fields with a response over 10% increase in productivity. You've got a whole range of fields here which are intermediate. You either get no increase in productivity or a relatively modest increase, which sort of maybe is 10% or less. And then you've got some fields where you've got low mycorrhiza growth response with no really or deleterious effects on growth of the crop by adding the inoculum. So there is really potential, a lot of interest in using these mycorrhizal fungi to promote crop yield. So this is why it matters. Okay, and in fact, you can see this. If you go to any garden centre, you can actually find inoculum of mycorrhizal fungi. And you can see that there are some crop production systems, particularly controlled crop production systems, where mycorrhizal fungi are being added to cropping systems. And also some interest in growing these, adding these to crops at the field scale as well, or at least modifying our rotations, the way that we grow our crops, reduce tillage, reducing the number of brassica in the rotation, because brassica are non-mycorrhizal crops that reduce inocular mycorrhizal fungi, reducing our nutrients and increasing our dependence on mycorrhizal fungi. But not all mycorrhizal fungi look the same, so we have no idea about the functional diversity of arbuscular mycorrhizal fungi, literally nothing. We don't know what those different fungi do. We just know broadly that they're grouped into a class of fungi that promote crop growth. Now, over the years, it's become apparent that there are two morphologies of arbuscular mycorrhizal fungus. One is which we call the coarse mycorrhizal fungi, and this is the predominant fungus that we've studied over the years. We know these are formed by glomeromycota because we can amplify them. We have PCR primers to amplify them. These are the mycorrhizal fungi that are used in our crop inoculants because they produce very large spores that we can characterize. And pretty much all of the understanding of the biology of mycorrhizal fungi comes from these, what are called coarse glomeromycota fungi. But lurking in the background, there's been this particular morphotype called the fine root endophyte, and it produces a very, very small hyphae, very small spores, strange hyphal structures. This is what it looks like. It's been known as glomus tenuis. And I just want to show you a sort of a video that my student Josh Cole took through a root. This is now a root that's been stained up with a fluorescent stain, and we're starting up halfway through the root. And I'm going to show you what happens as we move through the root, what these fungi look like within the root. So as we move through the root here, you can see we have these structures which are within the root. You can, oh, what's up on there? You can see these structures which are becoming more dense within the root. These are arbuscules of this fine root endophyte. And as we move on through, you can see we're moving on through the cell. And then as we come out of the cell again, we can see sort of fungal hyphal structures within the root as well. Very different to tetracladium. Tetracladium on the outside of the root, this particular fungi on the inside of the root forming arbuscules, which are involved in carbon exchange, nutrient exchange between the partners. We did some work with collaborators at the University of Western Australia, Meg and Ryan, and we actually showed what we did is we sequentially enriched these fine root endophytes by sieving out the coarse spores of glomeromycota, which are very big. We can sieve them out using spores. So we enriched the very small spores of fine root endophyte and then grow plants in that inoculum. And we found that as we increased the composition of fine root endophyte, this composition of morphology of fine root endophyte, as we increased the proportion of fine root endophyte morphology, we actually reduced the number of sequences that we had of glomeromycota and increased the number of sequences that we had of this fungal group, Mucoromycotina. So really this was indicating really that these fine root endophytes are not formed by glomeromycota, they're formed by Mucoromycotina. And so really this is everything that we know about mycorrhizas and arbuscular mycorrhizas. We've only been studying one part of the symbiosis. We've missed this fine root endophytes symbiosis. We know nothing about it. And so over the past maybe 5 or 6 years, we have had a number of projects, Megan and I, to study the ecology of this group, trying to understand how they interact with plants in the environment. And one of the approaches that we've done to do this is to analyze its genome. And I want to tell you very briefly about some of the work that we've done to do this. Now, this is very challenging. So if you can imagine, these organisms can't be grown. Tetracladium was easy. We could isolate it, we can extract DNA, we could sequence it. Now, this organism, we can't do it. And so now what we did is we used that metagenomic technique that I told you about earlier to assemble its genome. This is the project that we had funded by the NERC. The way that we did this is we took those samples of roots, we extracted DNA from them. Now you can imagine that the DNA extracts will include DNA from the roots, the bacteria, the protists, any fungi that we have in the system, as well as fine root endophytes. And then we did shotgun sequencing. So we broke up all of these different bits of DNA from these different organisms into little fragments. Now what we then need to do is we need to assemble them. And we assembled those reads into what are called contigs. And then we looked at those contigs, so basically overlapping regions of DNA, like putting a big jigsaw together. We were able to bin them. And so add those contigs together to form parts of genomes of different organisms. And then we could look at the identity of these different bins to actually work out which ones were fine root endophyte. And this is work that we did with Chris Quince at the Earlham Institute. He's a global expert in bioinformatics of metagenome assembly of genomes. And then once we had that MAG, it's called a MAG, a metagenome assembled genome, we're able to do comparative genomics. What genes does it have? What does that tell us about the symbiosis? And we were able to do it. This was a pretty amazing thing that we were able to do. We just published the paper just before Christmas. We were able to look at its genome, its completion, its proteins that it had in its repertoire, and we're able to compare that to other fungi. And so we're able to compare that to about 280 other fungi in terms of a comparative genomics analysis. Now, very quickly, just tell you about what that genomic comparison looked like compared to Glomeromycota. So the genomic analysis told us, first of all, the symbioses act in the same way. Both miss fatty acid synthases. They cannot make their own fatty acids. Both cannot make their own thymine. They're obligated dependent on their host for fatty acids and thymine. Second, their arbuscules work in exactly the same way. We found there was a huge number of calcium transporters, more than any other fungi in that 280 list that we looked at. It's a particular signature of arbuscular fungi. And so this works by depositing calcium in the arbuscule at the end of life to stop it working. And then we're able to look at its lifestyle. So we found that our fine root endophyte had a complete B6 pathway. And this is really to do with the way that it interacts with the plant as it grows through and colonizes the plant and as it responds to environmental stress. The Glomeromycota don't have a complete pathway. They're much more dependent on their host for response to stress, and indicating they colonize the plant in different ways. Also, the fine root endophyte has a complete cellulose degradation pathway. They're able to degrade fungal cell walls, so they're more saprotrophic than Glomeromycota. And they also have, remarkably, a low abundance of transporter genes. So although we think of Glomeromycota as being involved in transport of nutrients, there was no particular genomic signature of fine root endophytes to suggest that they do this. So less developed specialism in nutrient transport. So that's the end, really. So really want to sort of conclude that microbiomes can be major determinants of plant performance. There is great diversity and complexity within rhizosphere microbiomes. Lots of factors interact to shape who lives in the microbiome of plants. When we look at the taxa we find within the microbiome, for most of those things, we just don't know what they do. But new genomic approaches like metagenomics is providing us with huge opportunities to try to unravel that functional significance. And there's still much new biodiversity to be discovered, including for groups that we thought we knew a lot about, like these arbuscular fungi. And I'll leave it there and just really would like to thank some of the people involved in this, particularly Sally Hilton at Warwick University, involved in the yield decline work, and collaborators at Rothamsted, Northumbria University, CFAS, for the work on tetracladium, Anna Lazar and Chris Quince involved in that work. And then for the work that we did on the fine root endophyte, Josh Cole, George Muscott, collaborators at Western Australia, University of Gothenburg and Earlham Institute. And of course, lots of funders for this, BBSC, NERC, Australian government funding, DEFRA, Medical Life Sciences Research Funding, Centre, which was studentships that we had from Centre, and MRBTP as well. Thank you very much.
Wow.
That's it for me.
Brilliant, Gary. Thank you very much indeed. A huge amount to think about. There's one question in the chat, which is George Tirado. I don't know whether you want to ask it in person or shall I ask it? I mean, just turn your microphone on if you want to ask it. No? Okay. So, yeah, the question is, Gary, what was the relation between soil pH and a higher number of fungal pathogens found in short rotation crops?
So pH was important at shaping those pathogens. Off the top of my head, I can't tell you which pathogens responded to that pH. But you know, there will have been a response of those pathogens. Yeah, I can't tell you what that exact relationship was.
Thank you. Any other questions? Yeah, sorry. Any other questions for Gary? Oops. Yeah. I think that's, oh, here we are. So one from Gary Bradbury. I'll just read it. Can you tell us, are there any other research groups around the world that have looked at just one crop and sampled many different crop fields over wide geography and compared with yields?
I think this is a sort of an area that is emerging. There's lots of different ways in which this can be done. In terms of approaches similar to ours, I mean, there are various ways of doing this. So there are, I mean, it's an emerging field, and in terms of the way in which people have done this and the approaches that people have used to look at the microbiomes, the complexity of microbiomes, whether they're focused on, particular groups or the more holistically within the microbiome like we did, there's a big field. So yes, if you're interested, Gary, I think you would have to do a literature search to find out more about this. But certainly within the UK, there's been some work recently on wheat, for instance, and looking at wheat crops, and sort of particularly looking at how things like crop varieties then interacting with the microbiome. In terms of landscape, there are fewer of those that look at landscape level. A lot of that work is at more individual field locations and trying to maybe manage factors like crop management factors within fields and within a one particular location, rather than looking at a landscape scale. And that was the particular unique thing about this work when we did it at the time. Just
Right.
looking for oxen organics. Did we run tests? Not in this particular work, we didn't. But we have done a lot of work on organic farms with our arbuscular mycorrhizal fungi. And yeah, I mean, for the mycorrhizal fungi that we demonstrated many years ago, that organic farms are actually enriched in mycorrhizal fungi. We haven't particularly run these particularly more sophisticated techniques in terms of looking at community composition to try to find out what that diversity looks like. But that's certainly quite important, I think.
And then, thanks, Gary. Miriam's got a hand up. So would you like to ask a question? Oops, you might
Yes,
have
good
to
afternoon.
Oh, okay,
Yes.
carry on.
Hello, good afternoon. Excellent talk and very impressive results. Thank you very much for the talk. I was wondering, so in some of the experiments, it was analyzed, the microbes, the interactions that were already present on the soil. But you mentioned, for example, an experiment done in Switzerland in maize, and there a mycorrhiza was added. And my question is, does it matter for the experiments whether the microbe is it native or it comes from other areas? Thank you very much.
Yeah, this is quite important. So I would say that the problem with inoculation is that quite often inoculants do not survive within the soil you add them to. That is the huge issue with inoculants. And so that's one of the reasons there can be huge variability within locations for the performance of inoculants. And so you can see in that particular experiment, you notice that they looked at, they didn't look at crop yield. This is about productivity, it's growth of the plant, first of all. And I think, it's quite important when you do experiments like this that you get the right metrics to answer the questions that you're interested in. So there are regulatory requirements in terms of your organism if you're adding it back to soil as an inoculant. And I'm not an expert in this, but I do understand that in the UK you have to show that the organism you're using actually is native to the UK. I don't think it matters if it comes from overseas as long as there's evidence that it occurs in UK soils. But yes, that issue about whether it's native, whether you've picked it up from that soil itself is important. But I would say that all inoculants can be quite hit and miss when you add them back to the soil that you originally obtained it from, or whether you add it back to a different soil in terms of whether it will persist, whether it will then colonize your plant, and then whether it will give you any impact and the intended impact for which you added it.
Great. Are you okay to keep going, Gary? There's some more.
Yeah, I'll carry on. Fine. Yeah.
Okay. So, well, you can probably read them yourself. So one is much work being done into investigating which beneficial fungi are most effective in outcompeting or having a negative effect on those harmful fungi pathogens generally?
Yeah, so we haven't. So you know, in terms of the work that we did with it, we took it as far as you can see with the tetracladium. We haven't been able to take it so far into direct work to look at inhibition interactions between the tetracladium and the oilseed rape. But you know, of course, this is an emerging research area, and there will be groups that are working on this globally. So yes, this is absolutely something that's a research topic that people are working on at the moment, and particularly, you know, interesting that people are working on things like um, um, trying to work on mechanisms, trying to work out how these interactions are taking place, so you can sort of maybe bioengineer those interactions, right through to sort of more agronomic approaches to sort of add inoculants back. So lots of fundamental and applied work in that zone.
Great.
Okay, farmer shoes in the home county.
Yeah, farmers' shoes.
Well, I think the evidence is there. So you know you can do a quick ready reckoner, and you can work it out from the work that is published. All of this work is published. It's published by, you know, it's AHDB work, that original stuff from NIAB TAG. Our work indicates that the loss in yield that you see from our work at the landscape scale is exactly the same in the loss of yield that we were getting in those controlled experiments. I would say that at the landscape scale, you can see there was lots of variability. So, you know, you can see that most farmers were growing their crops in a 1 in 4, or 1 in 3 or 1 in 4 rotation frequency. And there was a lot of variability in the yield of those crops. So there's lots of site-specific variability. But, you know, if you are growing in short rotations, you know, you really shouldn't really be growing it in monoculture. I think from some of the DEFRA evidence, there is some evidence of that from some of the crop rotation data. Shouldn't really be growing alternately. 1 in 3, you're still going to be getting crop yield loss, and you'll even be seeing that signature at 1 in 4. So you know that's, you know, you have to think about that, and what that yield loss would be versus. I know that farmers take all sorts of factors into consideration for the rotation frequencies, but that's maybe something people, you know, need to think about. And remember, it's not just happening for oilseed rape; it's happening probably for lots of your crops. Okay, looking at that next one, David Talbot. That was George's question, really. It's about specific interactions between pathogen and soil pH. So, you know, I have to say that, you know, we were using commercial, these were commercially managed crops. And, you know, I think that most people will try to maintain their pH within a particular zone. I can't remember what the range of pHs was, but it was relatively narrow pH range. But you know, you're still seeing an impact of that pH range on the microbiome. And you know, I can't tell you off the top of my head what those interactions were with specific pathogens, but you know, they were definitely linked to pH. And in fact, when one of the slides that I showed you that I didn't really want to go into that detail with the pH thing, is just illustrating that huge impact that pH has on the microbiome, which is really good indicating for, you know, really. You tend to get fungal communities favored by low pH, and so really trying to have a sort of high pH will reduce that that sort of fungal component.
Great. So I think we've probably got to the end of the questions. So Gary, thank you so much. That was, I think that was an amazing talk, and lots and lots to think about. And we have been recording it, so hopefully it'll be okay, and we can send the link out. So thank you everybody for joining as well with the questions. And just to say, the next webinar is now lined up. It's on when it is February the 23rd, and Catherine Bradshaw from the Met Office, she's going to talk about the work that they're doing on how climate change is altering pest risks, and looking at modelling climate projections. And she's also, I've asked her if she'll talk a bit about long range climate forecasting as well, which might help people to anticipate periods of sort of high pest and disease risk. So again, thank you very much, Gary, and thank you all for joining, and have a good evening.
Thanks, everybody.
Actually, you could turn your camera on now, Gary, if you want.